What are the key components involved in homeostasis? It's often difficult to understand the concepts of homeostasis when we're focused in on physiological values. And so physiological measurements like solutes within the blood, solutes within a urinalysis, even things like pulse and blood pressure and respiratory rate are going to be examples of physiological parameters, of physiological variables that are going to be under the control of the body, maintaining some level of homeostasis. And it's difficult to view that in the abstract. And so we're going to try to look first at a concrete example that hopefully many of us are familiar with. And so we're going to think about being in a room over here. Within the room, we're going to have a thermostat. The thermostat is going to be set at 72 degrees. So 72 degrees is going to be the set point. And once that thermostat is set, we really don't have to think about maintaining kind of a comfortable temperature within the room. The thermostat is going to regulate the temperature in that room so that if it starts to get a little bit too warm, it's going to turn on the air conditioning and cool it off. If it starts to get too cold, it is going to turn on the heater and generate more heat and bring it back up. And so in essence, we're looking at homeostasis as a dynamic process. The temperature in the room is going to be oscillating a little bit around 72 degrees, in this case 72 degrees Fahrenheit. And that's all being controlled without us necessarily thinking about it. So how is it controlled? Basically, within that room, we're going to have a thermostat, and I'm breaking this apart as opposed to what a thermostat is originally. And so we're going to have a control center, which is basically the thermostat portion, which is going to be taking a look at the set point, comparing it with the input signal. We're going to have a receptor. In this case, the receptor is going to be a thermometer. And so with this thermometer, we're looking at it being 72 degrees. And so the thermometer is going to measure the temperature in the room, so measure the external stimulus. It's going to assess it at 72 degrees, and it's going to send an input signal into our control center, sometimes referred to as an integrating center. And basically what's going to happen within the control center or integrating center, it is going to compare the information from the input signal with the set point. In this case, if it's 72 degrees in the room, 72 degrees as our set point, we don't have to do anything. And so we don't need this output signal here. But what happens when the temperature of the room starts to decrease? And so we're going to erase a little bit here. And so what we're going to end up with is basically kind of poorly drawn, but the thermometer now is reading 70 degrees. And so 70 degrees is going to represent the conditions in the room, the value or the variable that we're looking at. And now we're going to have the receptor, our thermometer in this case, measuring the room temperature is 70 degrees. It is going to send an input signal to the thermostat portion acting as a control center or integrating center. And at that point, the control center is going to say, hey, we're supposed to be at 72 degrees, but we're now at 70 degrees. And so what we need to do is bring the temperature of the room back up towards its set point. And so what's going to happen is that thermostat is going to send an output signal to an effector. The effector in this case is going to be a heater. That heater is going to generate a response, and that response is, in essence, going to be more heat. The heat is going to be generated. It's going to accumulate in that room, and at a certain point, as that temperature accumulates within the room, we're going to see our thermometer going back up to 72 degrees. And so again, reflecting the external stimulus, the temperature within the room. When the thermometer reaches 72 degrees, it again is going to send an input signal, and it's a little bit more complicated than that, but basically the input signal is going to be coming from the receptor to the thermostat. Depending upon what homeostatic value we're looking at, sometimes the signal is sent, sometimes it's not being sent, but we're not going to worry about it at this point. We'll look at that with specific examples. So once we return back to 72 degrees, we now send our input signal, 72 degrees from our receptor, to our thermostat. The thermostat, acting as a control center, is going to compare the room temperature, our physiological variable, if we want to think about it like that, 72 degrees to the set point, 72 degrees. And at that point, we're again not going to have our output signal going out because it doesn't have to do anything. It's set where it needs to be. So this is going to be a good example of homeostasis. But instead of taking a look at this as an external measurement, we're going to take a look at this as a physiological measurement, and we're going to think about this in terms of blood glucose levels. Okay, so basically what we're going to do is modify our diagram, which we looked at the homeostasis of room temperature, in order to look at homeostasis of blood glucose levels. And so we need to change things around a little bit. Instead of taking a look at the room, basically what we're going to be looking at is going to be the blood. Instead of having a thermostat acting as our receptor, what we're going to have is going to be a pancreas cell. And we're not going to worry about what specific pancreas cell this is at this point. But it's basically going to be able to kind of examine the blood and determine what is the concentration or what are the levels of glucose within the blood. Now, this is making it a little bit more complicated, but instead of a thermostat with that set point, we're actually going to have that same pancreas cell. So in this case, the cell is acting as both the receptor and the control center. So this is the same cell. So the same pancreas cell. And instead of an output signal going to a heater, what we're going to be doing is going to other cells. And so those other cells are going to be things like liver cells, muscle cells, and cells that are active. And so basically cells that are metabolically active. And we're going to take this more heat out because we're going to take a look at blood glucose levels. Okay, so we're going to have this situation where we eat a big meal. So in the process of eating a big meal, we're going to bring in nutrients into our body. The digestive system is going to break those down. They're going to be absorbed within the small intestines. And then within there, they're going to be transported through the bloodstream. Well, we're basically going to see an increase in glucose within the blood. And so the pancreas cell is going to be examining the blood, evaluating the amount of blood glucose that is going to be, or glucose that's going to be present within the blood. In this case, we don't really have an input signal because the same cell is going to be acting as both the receptor and the control center. But basically what's going to happen is in a response to, in this case, an internal stimulus, the increase in glucose levels within the blood, our pancreas cell is going to examine it, determine that we have that increase in blood, and is going to make the decision in the control center kind of functions of that cell to send out an output signal. In this case, the output signal is going to be in the form of the hormone insulin. So that hormone insulin is going to be released by these pancreas cells. They're going to circulate through the body, and they're going to bind to receptors on our effector cells. And so it's going to bind to receptors. Insulin is going to bind to receptors on liver cells and muscle cells, and basically signal to these cells that we have excess glucose available within the body right now. And so take some of that extra and store it up. And so our liver cells and our muscle cells are going to take in those glucose molecules and then store them for a later period of time. It's also going to be, this insulin is also going to bind to receptors on metabolically active cells, so cells that are burning glucose, that are generating lots of ATP because they're metabolically active. They may be involved with building structures within the cells, they may be involved with producing lots of proteins, but basically they need a lot of ATP, so they need a lot of glucose. And so the overall response then is that our liver cells, our muscle cells, and our metabolically active cells are going to be using up glucose from the blood. And if we're using up the glucose from the blood, we're going to result in an overall decrease in blood glucose levels. And so that decrease in blood glucose, because these cells are using it, is going to kind of counteract or have a negative feedback interaction with that increase in the levels of glucose. And so what we've got here is an example of a good negative feedback loop. Okay, negative feedback is basically if it's triggered by an increase, the negative feedback is going to be a decrease in blood glucose level. And so in our example, if the room cooled, the negative feedback would be heating the room back up. And so what we're going to do then is, again, maintain our glucose levels at a dynamic level, near a dynamic set point. Okay, so our previous diagram really didn't look like what we're going to see in our textbook. And so we're trying to convert this into an example of a flow chart within the study of physiology. Okay, so we're going to take a look at it. Step one is going to be that we're going to eat a meal. As a result of eating the meal, digesting those materials, and absorbing those nutrients, we're going to see an increase in blood glucose. So increase in blood glucose levels, okay? That's going to be the internal stimulus. We're going to have a single cell here. So we're going to just draw this as a pancreas cell. And this pancreas cell is going to act as a receptor and as a control center. So it's basically going to kind of evaluate what is our blood glucose level in relationship to a set point. In this case, if the blood glucose level is higher than our set point, the cell is going to generate a response. And so that's going to be the output signal. And that output signal is going to be in the form of insulin. That insulin then is going to act on our effectors over here. So things like our liver, our muscle, and active cells. And these active cells are going to use up glucose. The overall effect of that then is that if we're using up the glucose, we're then going to decrease blood glucose levels. Okay, so as we decrease blood glucose, we are basically having an effect of decreasing or diminishing our stimulus over here. The stimulus was an increase in blood glucose level through the process of negative feedback. We are going to decrease our blood glucose levels back towards that set point. Once we receive, or once the decrease in blood glucose levels has brought our blood glucose levels back to its set point range, then our pancreas cell is going to stop acting. It's going to stop sending that insulin. And so this is going to be a good example of homeostasis. And a similar thing would be occurring by bringing up blood glucose levels if they are decreased within the body. And that's going to be a different hormone, but it's going to be the same basic type of negative feedback loop and homeostasis to maintain our blood glucose levels around an internal set point.